Good evening. I would like to thank for the invitation to present a seminar as a part of International Academy for Clinical Hematology. I'm Katia Beider from Sheba Medical Center, and I will share with you our current knowledge about the role of immune microenvironment in multiple myeloma. So let's start with a short introduction about the nature of myeloma disease. I guess we are all familiar with this beautiful figure describing the development of myeloma that arises as a result of complex and multi-step changes in the bone marrow and is preceded by precursor states such as a monoclonal gammopathy of undetermined significance, MGUS, and smoldering multiple myeloma. Multiple myeloma can progress to bone marrow independent diseases such as extramedullary myeloma and plasma cell leukemia. Primary genetic events in the development of MGUS and myeloma include chromosomal translocations involving the immunoglobulin heavy chain genes. And the number of secondary genetic alterations increases from MGUS to smoldering and then to multiple myeloma. And we also know that the interplay between multiple myeloma cells and the bone marrow microenvironment is a crucial for myeloma development, treatment, and progression. Bone marrow myeloma cells are highly dependent on neighboring cell signals for survival, which allows them to grow and proliferate. And we know that bone marrow microenvironment consists of multiple cellular compartments, including mesenchymal stromal cells, immune cells, different immune cells, endothelial cells, osteoblasts, and osteoclasts, creating a distinct milieu that supports immune escape and promotes progression of myeloma disease. And the growing number of studies, current studies, suggest that in addition to clonal evolution and strong inter and intratumoral heterogeneity of malignant myeloma cells, also immune microenvironment undergoes reprogramming during disease development and progression. And actually, the interplay between the immune system and malignant plasma cells is implicated throughout all stages of myeloma, including MGUS. At early stages, cytotoxic lymphocytes such as NK and CD8 T cells effectively eliminates malignant plasma cells. In the equilibrium phase, immune system mediates functional dormancy and prevents outgrowth of malignant plasma cells. And in active myeloma, malignant plasma cells eventually overwhelm the immune system, leading to the development of the immunosuppressive milieu. And this is the escape phase. Re-entry into equilibrium phase can be achieved by remission induction therapy, which can contribute to long-term MRD control in some patients. But the immunosuppressive milieu is re-established after relapse. And the concept that the immune system mediates surveillance of early tumors has been demonstrated in several models. Evidence for immune surveillance in myeloma comes both from murine models and from studies on patients with early lesions. For example, this study demonstrated that the immune system plays a critical role in the control of myeloma progression and the response to treatment. The group monitored PK-MYC mice that had been crossed with CD226 mutant mice over a period of 3 years and found that CD226 limits spontaneous myeloma development because this response against myeloma was CD226 dependent, was immune response against transplanted PK-MYC myeloma cells, and it was mediated both by NK and CD8 T cells. This CD226 is DNAM1, important activatory receptor, and it is negatively regulated by TIGIT, immune checkpoint molecule. So we can suppose that TIGIT inhibition can restore T cell surveillance. And indeed, in a preclinical, another study, preclinical murine model of syngeneic stem cell transplant for myeloma, it was observed that CD8 T cell exhaustion and decreased expression of DNAM1 correlated with tumor burden in mice with relapsed myeloma, while TIGIT immune checkpoint blockade prevented CD8 T cell exhaustion and provided successful immune control of myeloma. And these results support the role of immune system in tumor control and identify TIGIT checkpoint as a promising therapeutic target in myeloma. The concept of tumor extrinsic control on the growth of MGAS is supported by studies in humanized mouse models. This study, for example, evaluated the engraftment of MGAS and myeloma patient bone marrow transplanted into humanized mice. And it was shown that pre-neoplastic MGAS tumor cells exhibited progressive growth in mice in the bones, suggesting that the dormant nature of the MGAS cells in patients is actually regulated, at least in part, by tumor extrinsic controls, including the immune system. And despite the progress in understanding the molecular pathogenesis of multiple myeloma and in developing new therapies, there are still many important questions that have to be addressed. The identity of the myeloma cell origin remains controversial. Researchers revealed that cancer stem cells might be responsible for the development of multiple myeloma. Also, clonal evolution is also a key topic in myeloma research field, which drives tumor progression, chemoresistance, and relapse in myeloma. And of course, the mechanisms by which the bone marrow niche contributes to myeloma pathogenesis remain largely unexplored. And additionally, there are no reliable biomarkers available for the accurate prediction of responses to specific drug classes, limiting the ability to select a more personalized treatment. So, deep molecular characterization of both malignant cell heterogeneity and microenvironment composition provide better understanding of myeloma biology. And traditional methods include microarrays and NGS. But there are limitations for these methods. They require bulk DNA or RNA from a large number of cells, and they provide average information about cell populations. And rare cell populations or unique cellular states might not be detected by these methods. So there is rapid progress in single cell technologies and bioinformatics, and it allows to uncover novel findings. It includes single cell analysis at genomic, epigenomic, transcriptomic, and proteomic level. RNA single cell sequencing has been widely applied in the field of myeloma research. Provide the analysis of cellular heterogeneity, identifying new cell subtypes, distinct cellular states, and elucidating dynamic cellular transitions during tumor evolution and microenvironment remodeling. These single cell analyses are rapidly expanding, and the number of published studies investigating the bone marrow microenvironment in myeloma is growing very fast. And this is one of the first works. It analyzed immune composition in the bone marrow in patients with MGAS, smoldering myeloma, and overt myeloma, compared to healthy donors' bone marrow microenvironment using single-cell RNA-seq. And here the authors observed a significant alterations already at early MGAS stage with enrichment in NK T and CD16 monocytes in the diseased bone marrow, and the relative decrease in plasmacytoid dendritic cells, mature neutrophils, and CD14 monocytes and other progenitor cells. And it's important to emphasize that these changes they appeared very early on a pre-malignant state. Among T cells, this is the same study. Among T cells, the authors analyzed cytotoxic and non-cytotoxic T cell compartments and observed an early accumulation of gamma delta T cells and regulatory T cells in diseased bone marrow microenvironment. The results also show patient-specific heterogeneity in both gamma delta and in Treg cells, emphasizing the need in larger cohorts for the analysis. Additional early changes in myeloma microenvironment included defective antigen presentation by CD14 monocytes due to intracellular accumulation of HLA-DR. And this dysregulated MHC class II expression resulted in T cell suppression in vitro cultures. And in addition, the progression from MGUS to smoldering myeloma was associated with the loss of granzyme K memory cytotoxic T cells, leading to reduced immunosurveillance in vivo models. And also symptomatic myeloma was characterized by an increased interferon alpha response across all immune cell types, which has been shown to promote immunosuppression, favoring expansion of myeloma cells. Another study similarly used single-cell RNA-seq and mass cytometry. This is the study of Beider and colleagues, and they showed that precursor stages already harbor bone marrow microenvironment alterations compared with healthy donors, including very early changes in myeloid cells and they showed increased terminal effector differentiation and enrichment of stem-like T cells in MGUS. You can see here a decline in the expression of CD95, CD86, c-KIT, and CD155, an increase in PD-L1 expression on myeloid cells, and also the proportion of TCF1 high, TBET low cells was found to be significantly increased in MGUS, and it was observed in both terminal, central memory, and effector memory compartments. Another study analyzed changes in T cell populations across disease stages, and they used single-cell RNA-seq. It was a study with the 8 MGA7 smoldering myeloma and 10 myeloma patients, and they showed substantial alterations in the immune microenvironment of myeloma compared with its precursor stages. Symptomatic myeloma was enriched for CD14 positive and CD16 positive monocyte populations, memory B cells, CD8 effector cells, and it was the decrease in CD4 positive memory T cells and mate cells. It's a subtype of cytotoxic T cells. But there was no differential expression of exhaustion markers, such as PD-1, TIGIT, and LAG3 when comparing stages. And importantly, cell proportions were heterogeneous even across the same disease stage, emphasizing the need for larger data sets and also underscoring the existence of an individual immune phenotype, which is likely influenced by other factors than the aberrant plasma cells. Another important question is, can we distinguish immunophenotypic correlates in immune microenvironment in patients achieving sustained MRD negativity versus those who lost or never attained an MRD negative state during the first year of maintenance therapy? So, immune profiling was performed on blood and bone marrow collected from 23 patients with multiple myeloma receiving lenalidomide as maintenance therapy. the authors performed single-cell RNA-seq coupled with VDJ sequencing on PBMCs, site of mass cytometry of bone marrow, mononuclear cells, and PCR repertoire analysis of PBMC. And they found that patients with sustained MRD negativity had an increase in circulating helper T cells and reduced exhausted T cells and Treg cells. Treg cells significantly decreased in patients with sustained MRD negativity, while mate cells, naive CD8 cells, and Treg cells were significantly increased in patients with prior high-dose melphalan autologous stem cell transplant. Another study characterized T cell diversity in bone marrow from healthy adults and patients with precursor and full myeloma using single-cell RNA-seq and T cell receptor sequencing. Multiple myeloma patients displayed increased percentage of Treg cells, CD8 central memory cells, and CD8 effector granzyme K positive cells, together with a reduced frequency of CD8 granzyme B positive T cells. And in addition, the group analyzed clonal T cells and identified phenotypic hallmarks of large intratumoral T cell clones. Significant differences between normal, benign, and malignant bone marrow aspirates were observed in medium and large clones. In healthy adults, these were enriched in CD8 granzyme B positive T cells. And in myeloma, there was a predominant of CD8 effector memory granzyme K. And in addition, the subset of potentially exhausted CD8 positive tox positive T cells was detected exclusively in the bone marrow of 4 out of 10 myeloma patients. And the authors compared the distribution of T cell clusters among small, medium, and large clones. And in healthy adults, large clones were enriched in CD8 granzyme B T cells, again, gamma delta T cells. In MGA smoldering myeloma, there was a predominance of CD8 effector memory granzyme K T cells. By contrast, large clones from myeloma patients were enriched in CD8 granzyme K perforin positive cells, CD8 effector granzyme K positive cells, and again CD8 tox positive T cells. And indeed, these potentially exhausted tox positive T cells, which were uniquely detected in the bone marrow of myeloma patients, were particularly enriched in large clones. And these results suggest a potentially increased dysfunction of large T cell clones in myeloma. So they additionally analyzed these large T cell clones, and they found that these large T cell clones also show the co-expression of multiple immune checkpoints, including PD-1, LAG3, and TIGIT, suggesting, again, a potentially dysfunctional phenotype. So they combined, they used combined treatment with anti-PD-1 plus anti-LAG3 or anti-PD-1 together with anti-TIGIT in immunocompetent murine model of myeloma, and they observed longer increase in overall survival, longer overall survival, and delayed tumor growth. And another important observation in this study was low expression of CD27 in large T cell clones. CD27 is a well-known marker of T cell activation and antigen-dependent differentiation, and there was a significant correlation between the number of T cell clones and the ratio between CD27 negative and CD27 positive T cells. So the extent of a clonal T cell expansion could be estimated in larger series of patients with available immunophenotypic data, according to the CD27 negative, CD27 positive ratio. and it may be used as a surrogate marker of clonal T cell expansion. And in addition, the presence of CD27 negative, CD27 positive ratio above a 0.3 was significantly associated with longer progression-free survival in both transplant-ineligible and transplant-eligible patients, and demonstrated independent prognostic value for progression-free survival in patients. This is another study that explored the dynamic features of immune cells in myeloma bone marrow and the underlying mechanisms of immune cell dysfunction, again using single-cell RNAs. And here the authors found that besides PD-1, abnormal expression of two serine threonine PIM kinases, PIM kinases, KLRB1 and KLRC1 were involved in the defect of immune cells in myeloma patients. And these KLRB1, a high CD8 T cells from myeloma patients, they displayed lower interferon gamma abundance when they were activated in vitro, which supports the weakened cytotoxicity of these CD8 T cells. It is known that elevated expression of PIM kinases can induce metabolic defects in immune cells, and indeed, aberrant metabolic processes were associated with the immunosuppressive microenvironment in myeloma patients. Decreased amino acid metabolism promoted the dysfunction of CD8 cytotoxic T cells. And aberrant lipid metabolism was associated with the deregulation of NK cells in myeloma. And as was mentioned earlier, the disease course remains very heterogeneous, with some patients relapsing within months of treatment initiation and others experiencing deep remission for more than 10 years. And this observation has led to the hypothesis that myeloma cells can enter a dormant state with regrowth over time when conditions become permissive, particularly when immune surveillance weakens. So deciphering the alterations within the immune microenvironment during therapy and in relapsing disease is challenging. And this study investigated 20 relapsed refractory myeloma patients prior to and post salvage therapy and compared them to 8 healthy donors. And in this population, which was heavily enriched for gain1Q, the investigators found depletion of CD4, CD8 naive, and CD4 memory cells with the enrichment of CD14, CD16 monocytes, effector T cell populations like CD8 memory and cytotoxic cells, and PD-1 positive gamma delta T cells. With an increase, also they found in addition to these findings, they also observed inflammatory changes in bone marrow that were primarily associated with a CD14 positive monocytes. So these inflammatory factors were mostly expressed by monocytes and by myeloma cells. So these were the changes of refractory myeloma in this study. Another study addressed the role of immune microenvironment in early relapse patients. In this work was performed again single-cell RNA-seq on the immune cells in patients with rapidly progressing disease, less than 18 months to progression, compared with those without progression within 4 years of follow-up. And early relapse patients had significantly higher numbers of exhausted CD8 T cells that express again granzyme K and TIGIT, with a decreased expression of cytotoxic markers like perforin 1, granzyme B, and granulin. And similar to the previous studies, there were also alterations within the monocyte macrophage compartment. Non-progressor samples show a much higher count of most myeloid subtypes compared to rapid progressor samples, higher enrichment of immature cell types such as granulocyte macrophage progenitors, these GMP cells, and CD1C positive dendritic cells. And in contrast, rapid progressors demonstrated an increase of M2 macrophages. And the analysis of cellular communication predicted the dysregulation in activation of pro-proliferative signaling pathways, including BAF, CCL3 and CCL5 signaling, and IL-16 signaling. And last but not least, recent studies investigated alterations of the bone marrow microenvironment during immunotherapies, as those heavily rely on functional immune system for optimal efficacy. And there are several immunotherapeutic strategies in myeloma field, including PD-1, PD-L1 checkpoint inhibitors that demonstrated disappointing results in myeloma patients, monoclonal antibodies, CAR T cells, and very promising B-specific antibodies. And regarding these B-specific engagers, there is a study that determined the immunological single-cell mechanisms of resistance to B-specific BCMA CD3 T-cell engager therapy. This novel immunotherapy with high response rates in refractory myeloma. Here, our authors analyzed the transcriptome T-cell repertoire of bone marrow-associated immune cells from healthy bone marrow donors, newly diagnosed myeloma patients, and relapsed refractory myeloma patients receiving BCMA, as I mentioned, BCMA CD3, B-specific antibody monotherapy. And their analysis show that B-specific T-cell engagers lead to clonal expansion of immune cell subtypes, particularly effector CD8 T cell. And the response to B-specific engagers is actually driven by CD8 T cells. You can see here the reduction in CD4 memory subset and increase in CD8 effector CE3-CR1 positive clones post-treatment with B-specific antibodies. And in addition, the abundance of exhausted-like T-cell clones was associated with clinical response failure. Clones derived from non-responders, patients with predominantly assigned, they were predominantly assigned to this CD8 exhausted-like tox cluster before therapy initiation, and the frequency of pre-existing CD8 exhausted-like tox clonotypes was further correlated with progression-free survival of patients treated with the B-specific T-cell engagers. And to assess if the abundance of these pre-existing exhausted-like CD8 T-cell clones was associated with a response to another BCMA-targeted immunotherapies, the authors performed a single-cell RNA-seq on the bone marrow resident T-cells of 9 relapsed refractory myeloma patients receiving anti-BCMA CAR T-cell therapy, but they did not find any prognostically relevant association of exhausted-like CD8 T cells in these patients. So this suggests that exhausted tox prediction marker in resident bone marrow is specific to active immunotherapies, such as specific T cell engagers. And importantly, treatment failure in this study or loss of response to B-specific T-cell engagers was not only associated with immune microenvironment alterations, but also with myeloma intrinsic adaptations, such as loss of the target epitope, BCMA, and MHC class 1 protein, downregulation of MHC class 1. So taken together, these findings demonstrate that two modes of action of the specific TCE treatment in multiple myeloma. There are two modes. The first is the expansion of specific transcriptionally defined T cell clones upon stimulation. And the second is differentiation, and the second is MHC class 1 dependent priming of naive T cells. And what about the challenges and future perspectives in single cell research of immune microenvironment in myeloma? So it's important to note that bone marrow microenvironment single cell data are usually derived from a random bone marrow site and might not be representative of the whole disease process. And this is particularly important for myeloma, as the vast majority of patients present with focal lesions where genetic profiles of myeloma cells have been shown to be distinct from the random bone marrow site. And indeed, first reports suggest that this is also true for the bone marrow microenvironment at these sites. For example, in this study, using single-cell RNA and ATAC-seq multi-region sequencing on paired random bone marrow and focal lesion samples from 17 newly diagnosed patients, the group analyzed myeloma subclones and immune cell composition, and they found that macrophages were significantly depleted in the microenvironment of focal lesions. But in T cells, proportional changes in T cell repertoire, but not site-specific expansion of T cell clones in intramedullary lesions was observed. So, in conclusion, single-cell technologies in immune microenvironment research have already influenced the myeloma field by identifying mechanisms, underlying treatment resistance, and they have the potential to further advance our current understanding of risk classification, prognostication, and individualized treatment approaches. A high costs currently limit their application, but it is anticipated that technical advances and broader use will significantly reduce the cost and allow for larger sample collection. And in addition, comprehensive and longitudinal profiling of the immune microenvironment using multi-omic single-cell approach with integration of genomic, epigenomic, and mass cytometry profiling of myeloma cells and immune cells will eventually lead to improved clinical outcomes and cure in myeloma. And thank you for your attention, and I'm ready for the question. Thank you.